Hey everyone, Brock here. Unfortunately, I haven't had much time to create some new videos, so I wanted to share with you one of the live classes that we did in which we introduced the kidneys, we had a look at the nephron, the functional unit of the kidneys, and sort of explored why it is they are so important in keeping us alive, happy, and healthy. Enjoy! So, before we had a quick 10-minute breather to, well, catch our breaths and wrap our head around things, we essentially just looked at this filtration pressure. And what's just important to note here is that we need to have a certain amount of pressure in order to, push these, this fluid, our blood essentially, through this glomerular capillary and into our nephron. Okay, this is the filter. This is sort of the filter paper of the body. Now, again, you don't need to know numbers. We don't need to know all of these specific bits and bobs. The most important thing is that just we understand the general process that is happening, which if we remember what is happening with bulk flow in our capillaries there, we're doing well. Now, the next part I want to look at here is essentially the juxtaglomerular apparatus, or what we are looking at here with our granular cells and our macular denser cells. Now, there was a slide back here. Here we go. And I said, I'm going to come back to this slide. This is what we're going to be focusing on now. So the big thing here is that we obviously have our afferent arteriole, blood's going through our glomerular capillaries and leaving via the efferent arteriole. Now, we can control the rate in which this filtration occurs. And this is called EGF. EGF. Wait, EGF? Oh my goodness, guys. My brain is just... It's having a bit of a TKO moment. Oh my goodness. Okay, it's GFR. Now, the reason... I should probably explain why my brain just had a bit of a seizure there, is my master's thesis, I explored a receptor and a signal, a signaling ligand called EGF, epidermal growth factor. And this is GFR. So yeah, my brain still sometimes just has a bit of a seizure during those, whenever I talk about those two. So GFR is glomerular filtration rate. And this is looking at the rate of filtration that is occurring in our glomerulus. And we can change or adjust that flow. Now, there are two main ways in which our body does this. The first one is looking at our, actually I'll talk about the granular cells first. The granular cells. Now, these are located within the afferent arteriole. And what these do is that these contain stretch receptors. So in the same way, like with our blood vessels, and we can sort of determine the amount of blood pressure we have by looking at the stretch of our arteries and our arterioles, we can do the same thing here with our afferent arterioles and our granular cells. And what they do is when they stretch a certain amount, okay, if they don't stretch a certain amount, if the blood pressure is high or low, we can control the flow into the glomerulus accordingly. So a good example there is, like, let's say we have a drop in blood pressure, and let's say the flow to our glomerulus here drops. So what we can do is, if we do have a drop in blood pressure and we wish to sort of increase that GFR, that glomerular filtration rate, we can actually cause some vasodilation to occur in the afferent arteriole. So by dilating those vessels, we're increasing that blood flow, getting more blood into these glomerular capillaries here, which will increase the amount of glomerular filtration. The second one here, this one's a little bit more tricky to wrap your head around, but we'll be okay, is the macular denser cells. Now, these macular denser cells are sort of located at the top of the ascending loop of Henle / the distal convoluted tubules. And what these cells do is they measure and detect the concentration of sodium. Now, what happens here in terms of our glomerular filtration rate? Let's say our filtration rate was really, really high. Okay, we're filtering heaps of stuff and we're going quick, 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 go, 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 go. And we're screaming through our proximal convoluted tubule and zoom, we go down a loop and up the loop and we're going really, really, really fast. Cool. But the problem is that if we're moving really, really fast, we're not absorbing things. Yeah, we're not absorbing as much stuff because we're doing it quicker. We don't have as because the substance is moving through faster. We don't have as much time to really absorb those substances. So what happens is if our filtration rate is really high, as we come screaming down our descending loop of Henle, our ascending loop of Henle, remember it's the ascending loop is when we're reabsorbing those sodiums and chlorines and those potassiums, all of these salts. If we get to the top of our ascending loop of Henle here, and suddenly these macula densa cells are going, whoa, this is still really salty. Okay, there's still lots of sodium in here. That's obviously a bit of a problem because it means, okay, we're moving through this too fast. We aren't reabsorbing things properly. We need to slow things down. And that's what I'm sort of explaining here. So we've got our afferent arteriole, we've got our macula densa cells. Now, granular cells, again, granular cells can go by two names. We can call them granular cells or we can call them juxtaglomerular cells. I personally call them granular cells. Now, what can happen here is that the macula densa will then send a signal here to the afferent arteriole and say, whoa, hey, we are moving way too fast. We need to slow this down. And what will happen is we will start to see vasoconstriction in the afferent arteriole. And that will slow down, or that will reduce the amount of blood moving into the glomerular capillaries there, which is going to reduce our glomerular filtration rate. So again, regulation of our GFR, our glomerular filtration rate, is twofold. We can break it down into intrinsic and extrinsic. Now, intrinsic control is what I've literally just explained. We have myogenic and tubular glomerular feedback. Now again, these seem very complicated terms. Myogenic control. This is just simply what I described with our granular cells here. This is just simply looking at the stretch of that arteriole and looking at the amount of stretch and the amount of flow that is happening in that afferent arteriole. So we can look at that stretch and we can vasoconstrict or vasodilate to control that flow accordingly. The second one is tubuloglomerular feedback. So within the nephron, we have these tubules, our proximal convoluted tubule, our, loop of Henle, and our distal convoluted tubule. So we have our macula densa cells communicating with our, essentially the glomerulus. So tubuloglomerular feedback, it's basically communication between that ascending limb, that tubular part, and our glomerulus. And this is where we can control the amount of blood flow going into the kidneys by looking at that sodium concentration. Because again, if sodium concentration is far too high, when we get to the top of the ascending limb of the loop of Henle, it means we're not absorbing stuff properly because it's moving through too quickly. If it is too low, then that's telling us, okay, well, it's like, we need to probably speed things up a bit. So we can control the ideal speed based upon by looking at that sodium concentration in the nephron. Now, we also have extrinsic control. So, looking at our extrinsic control, we have sympathetic nervous system. Now, this obviously has an effect because if we have our SNS activation, so let's say again, Trish comes out with a big stick and she's chasing me and I'm running for dear life, then what is going to happen there is that I'm obviously going to divert a lot of my blood flow away from the kidneys because I'm running for my life. My priority is not on creating urine. So of course, what's going to happen then is that the GFR is going to be significantly reduced. The second one is the renin-angiotensin-aldosterone system, so RAS. Again, I'm going to reserve that and I'm going to be talking about that next week. And the last one here is looking at sort of looking at paracrine endocrine hormones. So looking at sort of cortisol, looking at adrenaline and noradrenaline, all that sort of stuff. This can obviously control the amount of blood flow and the total amount of glomerular filtration that is occurring. So these next few slides is pretty much just what I've already explained here. So again, if we've got a change to our overall blood pressure, so if we, instance, have a very high blood pressure, we're getting a high amount of flow, we need to reduce that filtration rate. We can undergo this vasoconstriction and reduce the amount of blood flow going into the glomerulus there, reduce that filtration rate. So again, we've already covered that the tubuloglomerular feedback there, looking at measuring those that sodium concentration in the filtrate. We've got the sympathetic nervous system here. RAS, we're going to be talking about that later. Now, this lovely diagram here, I'm going to be bringing up this diagram again when we look at the end of next week, because this kind of ties everything nicely together, especially when we're looking at regulation of blood pressure and blood volume. What we have done is basically we've looked at these two over here. We're not going to look at this one just yet. That's next week's job. We'll deal with that then. We're going to focus on this side. And again, this is a nice little flow chart of what I was just describing before in terms of this myogenic feedback and tubuloglomerular feedback and controlling that glomerular filtration rate. So again, if we have a decrease in blood pressure, there's going to be this decrease in stretch, which means we've obviously got this decrease in glomerular filtration rate. So we get some vasodilation, increased blood flow, bing, bang, bomb, we've fixed the problem. Again, a decrease in glomerular filtration rate. This means that we're going to notice less sodium and less chlorine in the ascending loop of Henle by those macula densa cells because it's moving through way more slowly. That means that there's plenty of time to reabsorb a lot of those salts. And that's going to basically send some signals to the macula densa, say, hey, whoa, hang on, our sodium concentration here is too low. That's then going to send signals to our granular cells, looking at sort of the juxtaglomerular complex of the kidney, and basically say, look, we need to get increase that blood flow. So we're going to see that vasodilation there, with the result being an increase in glomerular filtration rate. So again, we're just looking at these two at the moment. This will be next week, and we'll tie it all together then. Now, holy dooly. So we've covered a lot of stuff. Yeah, we've done a lot of stuff. Now, this entire time, since we got back from the break, I should say rather, we are been here. Okay, we have been in the glomerulus. We haven't left. Okay, we've looked at the glomerulus and our afferent arteriole. So a bit of a reminder here again of our three primary functions. What are we doing here? First one, we are filtering. We are filtering things based upon size and obviously the rate of filtration. So that GFR, that glomerular filtration rate. The second thing is a lot of the things that are being filtered out, we do not want to get rid of. It doesn't mean it's waste. So we need to reabsorb it. So we can see our peritubular capillary down here. And the third point will be secretion. This is getting rid of stuff that our body doesn't want anymore, putting it into the nephron to be released as urine. Okay, so again, this is a good little summary slide here. We all should be familiar enough to be able to identify every single aspect of this nephron. We should be able to communicate clearly what each aspect of this nephron does and answer anything to do with this, basically. So we see here, renal artery, we've got our glomerulus, we've got our Bowman's capsule down here. We have our peritubular capillaries surrounding this. We have our proximal convoluted tubule, descending and ascending loop of Henle, our distal convoluted tubule, and our collecting duct, with anything that is being reabsorbed by these areas moving into our peritubular capillary, reconnecting back to our afferent arteriole. Afferent arteriole, going back into sort of our venous network, resuming that circulation there. Okay, so it's going back into circulation. It's not like all of these substances that we are reabsorbing are just sort of hanging out in the kidney, never to be seen again. Again, a lot of this stuff I've already explained. Oh, right. Okay. So big thing here to learn when we're looking at this reabsorption, okay, especially the key nutrients and minerals and stuff like that, especially things like glucose, our body is exceptionally good at it. However, that doesn't mean it is 100%. So when we're looking at the reabsorption of glucose, okay, we require a protein-based transporter there to move it out of the nephron. Now, the problem here is that that can be saturated. Imagine like you're going to, you're at Woolies or Coles and you're trying to buy some groceries and every single checkout lane is filled. That's what I mean by like trying to reabsorb glucose, but it's completely saturated. That means that your body is working 100%, like maximum speed, foot to the pedal, trying to absorb all this glucose. But if you're, say, someone who's diabetic, there's just too much there. It can't absorb all of it, which is why glucose doesn't, like all that glucose doesn't end up being absorbed. It goes all the way through. And hence one of the primary symptoms or the side effects there is, glucose in the urine, which is obviously very, very bad because obviously it's going to generate its own sort of osmotic gradient there and pull water out of you. So again, let us go through another quick breakdown of the nephron again, because again, I've stressed it to the moon and back, its importance. Now, these next couple of slides here are very, very good. They are also incredibly detailed. Okay, you do not need to know all of the detail on these next couple of slides. I'm just going to point out the key things that we need to be aware of. So again, we have our proximal convoluted tubule here. This is the big boy for absorption. So we don't need to know like, primary active transport via the basolateral sodium potassium ATPase pump and blah, blah, blah. Nah, don't bother about that. The big thing here is that we are just reabsorbing just everything that is super duper duper useful here. So we're grabbing sodium, we're grabbing water, we're grabbing vitamins, we're grabbing glucose, we're grabbing amino acids, we're grabbing any cations or anions. So cation is positive ion, anions negative ion. So our potassiums, our magnesiums, our phosphates, our sodiums, all of that sort of stuff. We're just rapidly trying to absorb as much stuff as we can to put it back into our circulation. Because again, if we do not reabsorb it, it turns to urine and is gone. Once we leave the proximal convoluted tubule, we enter the nephron loop, or the loop of Henle. Now, what we see here is a bit of sort of a cyclic function, right? So what's going to happen is we're going down our descending loop of Henle. And it's during this stage that we are absorbing water. Cool. How? How is it that we are absorbing this water? Well, the surrounding interstitial space here is very salty. So because it's salty, we've created that osmotic gradient and we are pulling water out. Okay, cool. That checks out. So then we go up the ascending limb of the loop of Henle. What does the ascending limb do? Oh, that's when we get the reabsorption of salts, our sodiums, our chlorines, and our potassiums in particular. Okay, so what does that do? Well, we are absorbing those salts, so sodium, chlorine, potassium, and moving it into the interstitial space, which means that the interstitial space is getting more salty, which, now that that interstitial space is getting way more salty, is going to help us reabsorb more water. So you can see how the fact that we're absorbing salts in this ascending limb helps us to absorb water in the descending limb. So they sort of work together in that regard. Now, again, I don't want you to go through looking at secondary active transport. I just want you to be aware that we are absorbing water and our salts. And the absorption of those salts is generating that osmotic gradient that is allowing us to absorb that water via osmosis. We then move into our distal convoluted tubule. Now, again, as I said before, once we enter the distal convoluted tubule and the collecting ducts, these are the only two parts that we have actual hormonal control over, which again, we're going to be talking about next week, but we can control the amount of sort of sodium and water recovery that we have from the distal convoluted tubule and the collecting ducts. And that can be done via aldosterone, antidiuretic hormone, and of course parathyroid hormone. Now, if we haven't gotten all of that absorption done already, it's okay. We have a last opportunity to do so, and this is via the collecting duct. Now, this does depend on aquaporin. Again, we're going to talk about that next week, but this is what allows us to have a bit of a say, or to help control how concentrated our urine is. So for instance, if I've just chugged down like 2 liters of water, chances are it's not going to be going like super duper active here in terms of like, you know, that like absolutely hyperactive water reabsorption, because I'm well hydrated. I don't need to reabsorb every single drop that's happening there. I can actually lose some of that water as urine and help sort of remove some of those waste products. If, however, I haven't drank anything in like, you know, a couple of hours, and I'm starting to get very dehydrated and my blood volume is starting to decrease, then my body is going to really start to activate a lot of mechanisms to really allow me to like hold on to as much water as I can. Okay. So the last thing now is our tubular secretions. This is where we're actively moving things into our filtrate for the purpose of them going into our urine for removal from the body. We're removing it as waste. So although a major focus of the kidney is looking at, you know, reabsorbing this, you know, reabsorption of that primary filtrate, it's not the only thing. We are trying to get rid of waste products. Okay, we're trying to excrete it. One big one is obviously the selective excretion of some drugs. This is why, you know, a lot of drug screenings are done by a urine test or a blood test, because a lot of drugs are removed via the urinary system. We can eliminate some of these passively reabsorbed substances. I haven't really spoken about passive reabsorption. That's when things get a little bit squirrely. Again, we're just trying to keep things nice and simple here. Now, what we have here is we can control the amount of potassium and hydrogen here. So we have like an exchange. So we can exchange, like we can lose a potassium, gain a hydrogen, and we can control our blood pH in this way, because we can control whether we remove hydrogens or we remove bicarb, which allows us to control our blood pH. So if our blood is too acidic, we get rid of some of these hydrogens, okay? We come back down to normal. If our body is too alkaline, we get rid of some of these bicarbonates. We can just get rid of it by putting it in the urine. We get more acidic. And this is what I mean by our kidneys are exceptional at buffering and controlling the pH of our blood. So we go through all of the nephron. We've gone through the proximal and distal convoluted tubules. We've gone through the loop of Henle. We've gone down the collecting duct. We've done everything we need to do. From there, it is now urine. So I've exited the collecting duct. It is now urine. We normally produce 1 to 2 liters a day, but again, it does vary enormously depending on how much you have drank, your current health, what you're currently doing, what you've eaten as well plays a big role in that, all sorts of different stuff. Now, obviously the color and sort of the visual characteristics of urine changes depending on your level of hydration. It can be sort of completely clear if you're very, very well hydrated, versus, you know, if you are quite dehydrated, it can be sort of quite a deep yellow in color, which is due to urobilin, which is a byproduct that's formed by the breakdown of hemoglobin, which is quite interesting because hemoglobin is responsible for your poop being poop colored and your pee being pee colored. So really, you have hemoglobin to just thank for so much. Oh dear. Now, our urine is slightly acidic, okay? It typically bounces around a pH of 6, but again, it does change depending on your blood pH, depending sort of how you're going. You know, if your blood is overly acidic, you know, your body will try to remove a lot of those acids and your urine will get more acidic, or vice versa in terms of alkalinity. Now, obviously, urine has an odor to it, especially because we are removing urea and ammonia. from your body, and that's what we covered last week when we were before the mid trimester break rather. That's when we were looking at like gluconeogenesis and that removal of the amino group, that deamination, and you can take that amino group and essentially create urea. And that urea is one of the main components in urine. And that's how it sort of got its name. Here we go. So now we're looking at some of the components of urine. Now, urine is predominantly water. A lot of urine is water. It does, however, contain urea, as I just explained, from the gluconeogenesis, so turning like amino acid into glucose. We've got creatinine. I'm not going to talk about creatinine or creatine monophosphate because what we're sort of, it's sort of to do with like muscle breakdown and muscle building, that sort of stuff. Ammonia, and again from gluconeogenesis, uric acid that's coming down from our nucleic acids. Not going to talk too much about uric acid. Of course, we've got our excess electrolytes. You know, if we eat too much, like if we got fish and chips and we had too much chicken salt, which I know there's no such thing, is there? There's really, you can't have too much chicken salt, but you get what I mean. If we have an excess amount of salt, our body will obviously try to excrete that or get rid of it via urine. Now, this composition here of urine will change quite a lot depending on diet, depending on your health, depending on your overall hydration, all sorts of stuff. All this sort of stuff is relatively common. It's relatively normal. What is not normal is sort of finding things like glucose. We shouldn't be finding glucose in the urine because, again, our body has a remarkable capacity to absorb or reabsorb, rather, glucose. Blood. Please don't have blood in your urine. If you do, talk to someone, okay? Maybe book an appointment with the GP because there absolutely should not be blood in the urine. Same with proteins. The presence of proteins or blood in the urine can be indicative of kidney damage. Ketones would be present there in terms of like starvation, or some types of diabetes can indicate presence of ketones in the urine. And bilirubin is looking at sort of the liver. Now, I don't expect you to sort of memorize or know. I mean, it's good to keep these in mind, but this is a perfect example here of like where you will be going from 1808 NRS. So once you finish sort of the studies with me and you move on to second and third year, this is where you're going to take a lot of this understanding of these key body systems, like, you know, the heart and the lungs and GIT and kidneys and all that. And you're going to be using your understanding on how these systems work, but exploring when they don't work properly. So for instance, you know, if we're finding glucose in the urine, you know, possible causes diabetes. Now, if we're looking at things like erythrocytes, you know, it could be trauma. So, you know, a blunt force blow to the kidneys. Yeah, so these are some of the possible causes here. Now, a lot of these we can't determine just with the naked eye. Like you know, depending on the quantity, we might be able to see blood in the urine. Okay, you'd see sort of a color change there. Looking at leukocytes or pus in the urine, it'd sort of look a bit cloudy, such in the case of like a UTI, a urinary tract infection. But many of these other things, like, you know, bile or proteins or glucose and ketone bodies, we can't see with the naked eye. So what we would have to do is do a urinalysis. So do an analysis of urine. Now, we can do this again by visual observation. So as I said, you know, looking at blood and, you know, whether the urine is cloudy, that can give us some indication of what is happening here with respect to the urine. In many other cases, like looking at, you know, glucose or ketones and stuff like that, we will have to use a dipstick. And you would just look for a color change to determine presence of various substances. Now, you can use a microscope. You can use microscopy here. I mean, not all that much to my knowledge. I mean, like, you know, if you do see a slight color change and you think it might be blood, you could take a sample and put it under a microscope to see if you can see these red blood cells. You do not want to see crystals. Why is crystals in the urine super duper 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 bad? Hey, there it is. Hole in one. Kidney stones. This just sounds... yeah, I've heard stories of how hideously painful they can be. So yeah, we've got crystals, which we do not want in the urine. Another sort of chemical urinalysis is a pregnancy test. And what this is you are essentially, you pee on a stick and you look for the little line to appear. And what is measuring is the presence of HCG, which is human chorionic gonadotropin. And what this is essentially a hormone that is released during, successful implantation of the sort of the blastula to the endometrial wall, sort of signifying pregnancy. So that's how a pregnancy test works. And of course, various drugs that are removed from via the urinary system. Yeah, Brentha, that's it. Yeah, crystal urea. Now, this hormonal regulation of urine production, I am not going to talk about this today. I'm going to be talking about this next week because it links very, very, very nicely with control of blood volume. So that's when I'm going to talk about these hormones here. Now, why do we care about looking at urine? Why is it so important? Well, looking at diuretics, which I will need to double check this, but last time I checked, it was the top 2. Out of the top 10, rather, most prescribed medications in Australia, in the top 2 were diuretics. And they are administered for the treatment of chronic hypertension. So basically long-term high blood pressure. Because what a diuretic does is promotes the release of water as urine. It increases urine production. Now, if we wish to increase our blood volume, we release a hormone called anti-diuretic hormone, which increases water reabsorption. So a good way to remember this is alcohol, okay? If you go out for a big night with your friends, okay, you have 1 drink, and then you have 2 drinks, and then you have 4, 5, 6, 7, 8 drinks. What are you doing a lot throughout the night? You are peeing like a champ. Exactly. Why? Because it is a diuretic. It is increasing urine production by decreasing urine reabsorption. So an anti-diuretic hormone does the opposite. And that would happen during dehydration or low blood volume. You will be decreasing the amount of urine formation and increasing the amount of water reabsorption, which again would be in our loop of Henle, in our distal convoluted tubule, in our collecting duct. So, we have produced urine. Hooray, we did it. What then happens next? Well, that urine goes down that collecting duct, it goes down those renal sinus, renal pelvis, and will go down to the urethra. Once we move down the urethra, it will then travel down into the bladder, where it will be stored, okay, until an appropriate time to release. Let's say that. Now, thankfully, we do have voluntary control over our bladder. And what we have here is this urethral sphincter. So once we get to a certain quantity of urine in the bladder, it is going to trigger these transitional epithelial cells and with those stretch receptors to basically send a signal going, hey, bladder's full. We should do something about that. What you will then do is at an appropriate time, you will voluntarily relax this urethral sphincter, and this urine will then move through this urethra and out to the exterior. Now, those stretch receptors and that transitional epithelium is a big reason why those who have been pregnant or are pregnant feel as though they need to pee all of the time, because there is pressure being put on the bladder and the body will think, oh, those stretch receptors are being triggered, I must have a full bladder, when you don't. Now, another thing we need to sort of take into account here is the urethra and the urethral length. Now, how do I say this? Guys have it pretty easy. Let's just say that. Now, an unfortunate issue that you poor ladies suffer with by having sort of quite a short urethra is the presence of UTIs, urinary tract infections. Because your urethra is only sort of 4-ish centimeters long, there's really not much of a barrier there between the bladder and the external environment, which again makes it more prone to UTIs. Men, on the other hand, our urethra can be over 5 times the length of yours, so it's a lot more difficult for bacteria to really work their way up there. However, sort of turning a frown upside down is looking at catheters. The insertion of a catheter for females is not that bad. I mean, I don't want to understate it. It's still not very pleasant, but due to female urethral length being only sort of 4-ish centimeters long, it's not that hard to sort of put a catheter in there, sort of tap the tank, as it were. For men, on the other hand, it's a lot worse. It is a lot more difficult and a lot more painful to have a catheter inserted. Now, a lot of these structures here, we're going to be talking about that a lot more in week 10 when we get to sort of reproduction and development. Now, I'm not going to really get bogged down the details here with the mituation reflex. All I just want you to be aware, excuse me, all I want you to just be aware of is that, the bladder will stretch when it's full. Our brain's going to receive that signal. Brock is going to have a hiccup. And what you're going to do then is essentially the urethral sphincter will relax and urination will begin. And guys, that is it for week 8. So what have we done today? What have we looked at? So we've looked at the structure and function of the urinary and the renal system. We've looked a lot at the internal and external anatomy of the kidney. We spent a whole heap of time looking at the nephron. Hint, yeah. And looking at each part of the nephron and what it does, why it's important. We looked at glomerular filtration. We looked at some of the means in which we can control it intrinsically and extrinsically. We looked at tubular reabsorption and secretion here. We've looked at the main characteristics of urine. We have not looked at the regulation of urine concentration and volume because, as I said, we're doing that next week. And we have looked at the elimination of urine from the body.